Good afternoon, everyone. For those of you who don't recognize me with a tie on, I'm Mike Griswold, Dean of the College of Sciences, and I want to welcome you all to the 2010 Distinguished Faculty Address. This is an esteemed and historic tradition at WSU. This address is a major part of Showcase, which is a day-long exploration and celebration of the university's teaching, research, and scholarly and creative activity. It began this morning, and I saw many of you there at the poster session this morning, and it will conclude this evening with the Celebrating Excellence Recognition Banquet. And now I would like to introduce Provost Warwick Bailey, who has some additional comments, and will introduce the speaker.
Thanks very much, Mike. Good afternoon, everybody. Just to repeat what Mike said, I'm not Elson Floyd. He had hoped to be here right now, but is in, I guess, en route from the Tri-Cities and certainly hopes to be joining us before the completion of this very important institutional event this afternoon. As Mike said, in many respects, today is the culmination of a very important week within the university's annual calendar. Earlier this week, we had one of a series of innovator presentations from one of our highly decorated or accomplished faculty members. On Wednesday, we celebrated the accomplishments and recognized contributions of many women from various sectors of our university, and today, we have, if you like, three different events. First of all, there was the poster session this morning, in which I think there were over 330 research and creative activities represented collectively. In total, I think there were 770 faculty and students' work were reflected in those 330 plus posters. So clearly that is an activity which covers the broad expanse and reflects the very, very diverse array of scholastic and creative activities that we associate with our Research One university. Just to elaborate a little bit more, and I hope that many of you did get up there, I commend Fran McSweeney and her committee for the efforts they did in selecting and organising this morning's session. There are 195 graduate students and an additional 76 undergraduate students who collaborated with the faculty and whose work was on display earlier this morning. And then, as you know, and I hope many, if not all of you, will be there this evening, we celebrate excellence, widespread excellence in all shapes and forms, as it takes place on our 4 campuses and our multiple other locations located through the state of Washington. And it is appropriate, I think, that we take time, especially when times are tough as they are, to reflect upon, recognise and applaud accomplishment and be appreciative of the many shapes and forms that such accomplishment in such ways that it's represented. Certainly throughout Washington State University's history, research, scholarship and creative activities by our faculty have played major roles in the social fabric and economic health of our state, our nation and our world. Whether it's associated with the development of new varieties of wheat, cherries, tests of human and animal diseases, scholarly activity that challenges assumed methods and concepts, something that promotes a broader-based understanding of what I'd call the human condition. Washington State University professors certainly make a difference through their many discoveries. And so, as you are well aware, Showcase is really a celebration of these contributions, and I thank everybody who's really worked so hard to make it such a memorable and a meritorious day. While Showcase, and particularly the poster session and the evening celebration of excellence, have evolved principally in the last few years, one element the Distinguished Faculty Address is now definitely an established and long tradition at Washington State University, stretching back more than 50 years to its first delivery in 1958. And really, when you look at that list of people who have been asked or invited or honoured with the opportunity to present the Distinguished Faculty Address, it truly does read like a list of who's who at Washington State University. Leading researchers, scholars and teachers have all been lauded and recognised for their accomplishments by being invited to deliver this address. From the first three, who were Herbert J. Wood, Harold Dodgen and Claudius O. Johnson, who presented in 1958, until today, this honour does recognise exceptional members of the WSU faculty, and it's pleasing to see a number of previous recipients of this award here today to help recognise and celebrate this year's awardees' accomplishments. Because, as I say, these people are certainly accomplished in many respects, have achieved in many different ways. There are certain criteria that go into the selection of the Distinguished Faculty Address, selected or awardee. They include recognition at the national and international level, certainly from the perspective of being distinguished with regard to original contributions in one's discipline. Consistent receipt of extramural support, national and or international service, and achievements in both undergraduate and graduate education. And certainly this year's Distinguished Faculty Address awardee fulfills these requirements most admirably. I told her that she didn't need any introduction, so therefore she wasn't going to get any. But that would probably be unfair because she's an extremely modest woman. So I just need to give you a little bit more background so that you can appreciate sort of the status that we associate with this recognition. Dr. Nancy Magnuson is known to many of you. She certainly has achieved international distinction for her research career in immunology and cancer while serving at Washington State University since 1978. She's published more than 95 peer-reviewed articles and has lectured all over the world. Australia, Canada, Korea, India, the People's Republic of China, and other areas. And though much of her research has focused on cancer, the scope and quality of her work contributes to basic medical research and as well as in applied fields. She's also been extremely active in the classroom and has been course director for high enrolment 400 and 500 level immunology courses virtually every year since 1986. She's been an exceptional mentor for both undergraduate and graduate students and has demonstrated extraordinary commitment and effectiveness in this area that we refer to as outreach and public service, which is certainly the third leg of the tripod on which Washington State University stands. Nancy has been involved in public education and peer review of research for the National Institutes of Health, US Department of Agriculture, American Cancer Society, and the Department of Defense continuously. Let me underline that, continuously. I don't know how you've done it. Since 1986. And it is for this outstanding service as a complete, I'll say it, as a complete land-grant university faculty member that Dr. Nancy Magnuson has been selected to give the Distinguished Faculty Address of 2010. The title of this is Why Target PIM1, got it right, PIM1, for Diagnosis and Cancer Therapy. Nancy, I look forward to hearing what you have to say. I congratulate you personally and on behalf of the institution for everything you've done, and congratulations on this selection. Ladies and gentlemen, Nancy Magnuson.
This should work. Is it on? Wait. Oh, it's not on. Wait. Okay. That's better. All right. So what I want to do today is talk to you about the research that I've done over the past 20 years. And it's about the protein called PIM1. And I'm going to try to convince you by the end of this talk why we think PIM is going to be very useful for cancer diagnosis and treatment. But before I do, I want to give you a little bit of my philosophy. And first of all, this is that exploratory science can be fun. Now, what I mean by exploratory science is investigating new, important developments. And there's a problem with this, actually, and that is that you could end up investing a lot of time, a lot of effort, and a lot of money, and you might not get what you were looking for. So that means that doing this kind of research also has a lot of risks. Because, for example, you might not turn out so happy with the results. If you're going for the gold, or in this case, the cheese. So, actually, to be successful, it takes a couple things. And one of the things is you have to be clever. But not only do you have to be clever, but all the people that work with you have to be clever. But I think equally important is that you need to be, it's not coming, lucky. And this is my opinion, and I'm sticking to it. All right. So now to get in with what I'm going to tell you about. So some points I want to cover in this next 45 minutes. First of all, I'm going to tell you how cancer begins. And the reason for that is to get everybody on the same page. Then I'm going to tell you why we think PIM is an unusual oncogene. Then I'm going to explain to you what we know about PIM. And 20 years ago, when I started on this, we knew absolutely nothing. So this was a risky thing to go into. And I'm going to try to explain to you how PIM contributes to cancer. And then finally, I hope I will have convinced you that PIM makes a good therapeutic target. Okay, how cancer begins. Well, cell division. Cell division is very important, and it involves precisely controlled events. And these events are dependent on the proper levels of proteins. If those protein levels are altered, what results is unregulated growth, and that's what we associate with cancer. The human genome contains at least 25,000 genes, and all these genes actually code for proteins. That's a lot of proteins, all right? However, it turns out that only a very small number of these proteins appear to be important for cancer production, maybe a couple hundred max. So when these genes are damaged, either by mutation or being rearranged, or even just simply being overexpressed, those gene products function abnormally. And what causes this damage or these mutations? Carcinogens. You know them as, for example, insecticides, herbicides. Environmental factors such as sunlight. Viruses and bacteria. Epstein-Barr virus that causes Burkitt's lymphoma, for example. And then probably maybe the largest contributing factor is chronic inflammation. So this small number of proteins, these couple of hundred proteins, falls into two groups. One group is involved with proliferation. The other group is involved with preventing cell death or involved somehow with cell death. So proto-oncogenes is the term that we give to the normal versions of these genes. That means they have the potential to become oncogenes. And the oncogenes then are the mutated genes or altered production of these gene products. So now let's switch gears and talk about PIM and its relationship to cancer. PIM was first discovered by a molecular virologist in the Netherlands, Anton Burns, and he was studying why Maloney-Mirren leukemia virus, which looks like this, when infecting mice, can actually integrate into the host genome and induce tumors. So the problem is that this virus does not carry a gene that causes cancer. So what he hypothesized was that it must be the genes nearby where the virus integrated. And that's when those genes near the virus were activated by the presence of the virus, that's what causes the tumor. And that's what we get, tumors. So what does PIM1 stand for? You'll see why we call it PIM1. Proviral, and the provirus is the viral genome that integrates into the mouse genome. Integration site of Maloney-Mirren leukemia virus. See why we call it PIM1? And this is site 1. Okay, so what is PIM? It's a serine threonine kinase, and that is an enzyme that puts phosphate groups on its target proteins. It's highly conserved. That means if you looked at mice, humans, cattle, the protein product is the same. There's hardly any difference in the amino acid sequence. Because of that conserved evolution, that suggests that it must be very important. So my laboratory actually determined what the sequence was. This is the amino acid consensus sequence, which the PIM gene recognizes in proteins and phosphorylates. And it is this serine and or threonine, one of them will be in that position, will be phosphorylated. So what is the consequence of that phosphorylation? Well, if you looked at the amino acid residue side chains, you'd see that you'd have this series of positive charges, and then there would be no charge flanking the serine or threonine. But when that serine or threonine is phosphorylated, it puts a negative charge on it. So what that does is it either changes the function of that protein that PIM phosphorylated, or it changes its cellular localization, or maybe even its structure, which would change some aspect of that target. The other important thing that I need to tell you is that PIM is constitutively active, and what that means is the enzymatic activity is always on. Now, we have lots of kinases in our body, but it turns out that almost all of them have to undergo some activation event for them to have enzymatic activity. So this is truly very unusual. So the experiment that actually demonstrated that PIM promotes cancer development was done by Anton Burns. And what he did was make PIM transgenic mice. Now, how he decided that PIM was the gene that was causing tumors in the mice that are infected with the virus was that he went into the tumors, he found where the virus was, he sequenced upstream, downstream, got those sequences, and then looked for the message that was being expressed in the tumors. And he found that in all the tumors, the one gene that was overexpressed all the time was PIM. So he said, okay, we want to see just how potent is this particular protein. So he made the transgenic mice, and then in the experiment, they looked at the incidence of lymphoma, a tumor, at the end of 3 months. Well, if you just took normal mice, put them in a cage, and looked to see how many of them come down with cancer, essentially none do. If you take normal mice and infect them with the virus, after 3 months, about 10% come down with tumors. And that's not necessarily a very high number. If you take the PIM transgenic mice and you look after 3 months, it's shocking, only 10%. This is not what they thought was going to happen. They thought, because this is expressed in all the tumors, it must be playing a major role in producing those tumors, but it's only 10%. This started to suggest something to them. So they went ahead and they did a couple more experiments, and the other experiment they did was they took PIM transgenic mice, infected them with the Maloney-Mirren leukemia virus, and what they found was 100% of the animals come down with tumors at the end of 3 months. They did the same thing with the transgenic mice and treated them with carcinogens, and once again, they come down with 100%. 100% of the animals come down with tumors. So, what does this mean? It means that PIM is supporting other oncogenic behavior of other genes, that PIM may not be actually inducing the tumor completely by itself. And it turns out when they go into the tumors and they look, although PIM will be expressed in 100% of the tumors, there are other genes that are expressed, and one of the genes that's expressed at a very high percentage is c-MYC. And it turns out that the expression of PIM is induced because there were mutations in it. So the virus actually had some effect on c-MYC in some of the tumors, and when they used carcinogens, they also induced mutations in c-MYC. And it turns out that if you took the oncogenic c-MYC and you put it into cells, it would kill the cells. It wouldn't make tumors. However, if you put PIM and c-MYC together in the cells, what you get is cancer that develops. All right. So what cancers have abnormally high levels of PIM? 20 years ago, we were all pretty convinced that the tumors that were really important that had high levels of PIM were leukemias and lymphomas. Well, since a lot of data has been accumulating on how PIM functions, people are taking a second look. And what they have found within the last 5 years that all these tumors have high-level expression of PIM. And in fact, one of the ones that's received the most attention is prostate cancer, and I will show you some more data about that in just a minute. So, what makes PIM an unusual oncogene? Two features. One, as I said before, it's constitutively active. Thus, that means that the absolute number of the amount of the protein is what's important. And the second is that the PIM gene is never found mutated in any of these tumors. So what's associated then is the fact that you have high levels of the PIM protein. So the following analogy I'm going to show you, I'm hoping will help visualize how we think PIM is contributing to cancer. And then after I go through this analogy, I'll highlight some of the most compelling evidence that supports this notion. Okay, so we start off with these unsavory-looking individuals, and they're bank robbers, all right? And if they're bank robbers, what they want to do is rob a bank, right? Okay, so they go ahead and they rob that bank. And that trips the alarm, and the police come, and the police stop them and put them in jail, and that bank is just fine. Okay? Now let's look at a slightly different scenario. We take the bank robbers, and we have the bank, but this time we have a getaway car. Okay? So the bank robbers attempt to rob the bank. It trips the alarm. The police come, but now they have a getaway car. And so what they can do is jump in that getaway car, and they get away. They've robbed the bank. You notice the money's gone. All right. Those two scenarios. Now let's put it in context of cancer. So we have our bank robbers, but really we're going to think of these bank robbers as good genes gone bad. And then we're going to think of the bank as your body. All right? And we're going to think of the police, in fact, as the DNA repair system is one thing, or a system for programmed cell death has evolved to kill off aberrant cells that are not doing what they're supposed to be doing. So the bad genes are trying to do something bad to your body, like start cancer, and your DNA repair system stops them. And if that doesn't work, then the programmed cell death gets rid of those aberrant cells. So that's good, all right? But now let's look at one more scenario. And that is, we have our bad genes, and your body that they're going to try to form cancer. And we have the protection here, but a different thing here. We have a getaway car, but this getaway car this time is PIM1. All right? So these bad genes try to cause cancer, and they trip the alarm, but because they have the getaway car, they're able to get away, and they cause cancer. So here's the take-home lesson. These genes did the dirty work. These are the ones that cause cancer. And that's because there's a mutation. For example, in c-Myc. You didn't know that's what he looked like, but that's it. PIM, on the other hand, helped them get away with it. So if you look at it, PIM is serving as an accomplice. Right? Now, let's go over some of the things that we've discovered about PIM that makes us think PIM is a good therapeutic target. And just remember, I'll reiterate, we didn't know any of this 20 years ago. So first, I just showed you that when mice are made to overexpress PIM, tumors do form, but only at a very slow rate. And second, the thing is, what happens if PIM is important? What happens if you don't have any PIM1? Okay? So here we have a mouse, and it has the PIM1 gene knocked out. So that means that these mice do not, under any circumstance, express PIM1. What happens? Essentially nothing. That's because the cells of the body are not completely dependent on it. There are other kinases that can compensate in instances. And in fact, this turns out to be an extremely good characteristic, because that means if you eliminate PIM, normal tissue should not be affected. That's a very key point. So then the question is, well, what the heck does PIM do? So one of the very first experiments that was done by a colleague of mine 15 years ago, Mike Lilly at UC Irvine, took growth factor-dependent cells, which are shown here. These cells cannot grow unless they have the growth factor present. So in this Petri dish, we have media, we have a couple cells, we add the growth media, and we watch and see what happens. Well, the cells don't die, but in fact, they even replicate. Now we take the growth factor away from these cells, and we look to see what happens. Well, the cells, they don't survive, but in fact, they even die. And so we see that we need the growth factor. So let's take a little twist on this experiment. And let's remove the growth factor from growth factor requiring cells. That's what we had in the first thing. That's this guy. But we're going to do something different. We're going to insert an extra copy of the PIM gene, so those cells are expressing a lot of PIM. And now we're going to look and see what happens. They're growing. They're not dying, and in fact, they're growing. And if we get rid of the PIM gene or the protein itself by adding an inhibitor specific for PIM, the cells die. And that's a good thing. So we conclude that PIM is acting like a survival factor and probably also having something to do with proliferation. But the question is, exactly how does this work? So now let's fast forward to within the last 5 years and look at some of the recent discoveries. PIM was found to be a potential clinical marker for prostate cancer. And the reason why this was even looked at is because the current protein that's used is prostate-specific antigen, or PSA. And clinicians don't like it because it's not really very reliable. And they're just very eager, and have been for the last 5 years, looking for a marker that would tell you whether or not that tumor is benign or is it really prostate cancer. So this work was done at the University of Michigan, and the goal was to identify proteins that might serve as diagnostic markers. So in the middle here, I have a diagram of cells. And this is the different stages. The first figure here is a cross-section of prostate tissue. And you can see you have a nice, this is a duct. You have epithelial cells that are lining it, uniform in size, nice open lumen. You go to the next step, which is really not cancer, but you see the cells are growing a little different. We go to the next step, which is full-blown carcinoma. The lumen is practically all filled. You have cells of all different sizes and shapes. And then you go to the worst of the worst, which is metastatic carcinoma. You have cells now leaving from the primary tumor and spreading out throughout the body. So what these researchers did at University of Michigan was they looked at groups of cells, and these cells would fall under cell adhesion, antiprotease, protease, etc., as you go around this. So what they were looking for were proteins that would be almost not detectable in normal epithelium, but as there was a progression towards the more abnormal cells, that an increase in these proteins would occur. And they did find two, and that turned out to be PIM1 and also c-MYC. Very interesting. Now, if they were going to use this as a clinical marker, on the next slide, what they would do is take a biopsy of the suspected tumor, and if the tumor was benign and they were staining for PIM, they would not expect to see any brown color, and they don't. However, in the carcinoma, you can see all these cancer cells in here, and they're really highly overexpressing PIM. So that says that PIM is very likely an excellent marker for prostate cancer. So how else does PIM contribute to cancer production? Well, PIM1 is part of the mitotic apparatus. Remember at the beginning when I said you need a little bit of luck? This is it. This was an unexpected discovery that one of my graduate students made. Now, the mitotic apparatus is a group of proteins that actually helps pull apart the duplicated chromosomes during cell division. And if PIM is overexpressed, it interferes with this mitotic apparatus, and it interferes with normal, proper cell division. So this was the experiment that was done. This is PIM in dividing cells. The green fluorescence that you see is PIM. The red is DNA. So my student signed up for this very fancy microscope that she wanted to use. She sets her cells up in culture. She wants them to grow, and her idea is she's going to fill up her plate with cells, and then she's going to go down and she's going to look and see where is PIM localizing in the cells. Well, it was time to go down and use the microscope, and she looked at her cells, and they hardly were growing at all. They were growing, but not very fast. So she had all these open spaces throughout here. And she was kind of not knowing if she should waste her time, should she just go down and go ahead and look at it, or wait and do it next week. So she said she was anxious, she wanted to do it. And she did, and to our utter surprise, we never expected to find PIM at what turns out to be the spindle pole of the mitotic apparatus. So she wanted to know if PIM was in the spindle pole. That might mean actually that it's associating with or phosphorylating some other protein in the spindle pole. It turns out there's 25 proteins in the spindle pole. So she had to go on the computer, look at all the sequences, see if there were any consensus sequences that PIM might phosphorylate, and sure enough there was. It was NUMA. And I'm not going to show you the experiments, but she did this, and it's published. She showed that PIM phosphorylates NUMA, and not only that, associates with it in a complex. So NUMA stands for nuclear mitotic apparatus. And this is a cell that is getting ready to undergo mitosis. The red is staining the mitochondria, so that gives you an idea of the cytoplasm of the cell. The spindle pole is at this end and this end. There's two of them in a cell. The spindles are these long, like finger projections that are actually attached to the chromosomes, which are stained in blue. So NUMA is at the spindle pole, and that's where PIM is too, and I'll show you. She did more experiments, and she showed by staining for PIM with green that it looks like PIM is in the spindle poles. We know that pneuma is, so she stained with blue to see where it was sitting, and it already looks like they're both in the same place. She stained the chromosomes, so she wanted to make sure that we might have something that looks like a mitotic figure. And then when she, this is, wait a minute, we missed a couple pictures here. Oh, too bad. Well, I'll just have to show you. There was a picture here, and they actually superimpose on one another. Sorry, I don't know why that happened. Take my word for it. Okay. Well, this leads us to another point, and that is that genomic instability and aneuploidy are hallmarks of many aggressive cancers, and prostate cancer is a very good example. So aneuploidy means that the cell has an unusual number or altered number of chromosomes. So here's our cell undergoing mitosis again, and I have a simpler diagram here to show you what is supposed to happen. We have the spindle pole here, and this green circle here is the spindle pole where pneuma and PIM are residing. The green sticks are the spindle, and they're attached to the already duplicated chromosomes that are ready to be pulled apart. And what should happen in a proper mitosis is that the chromosomes are pulled to the opposite sides of the cell, and the cell divides. So, in cancers, the spindle poles are often amplified when they should not be. In a normal cell that has not replicated its DNA, there'll be one spindle pole, but when the DNA gets replicated, another spindle pole will be made so that the cell will have two. In cancer cells, this direct link between DNA synthesis and spindle pole production is separated. And here you have a cell that has not yet replicated its DNA, but it has over-amplified the spindle poles. Why is this important? Because abnormal separation of chromosomes can occur. So here's a cartoon of a cancer cell that is getting ready to divide. It has duplicated its chromosomes, but the duplication of the spindle poles is no longer attached to the duplication of the DNA, so now we have extra spindle poles. And what happens? So when the cell undergoes mitosis, we actually get an uneven distribution of those chromosomes, and the daughter cell on the left has extra chromosomes. And why is that bad? Because those extra chromosomes could actually give that cell a growth advantage, and that is the first step probably towards cancer. Well, sorry, we just missed something, but so I'll have to go back on this. Oh, man. Okay, this was really beautiful, so I'll have to explain it to you. So in this row, we have a normal spindle and normal poles and normal DNA and a beautiful mitotic figure, okay? In cells that are overexpressing PIM, what you would have seen is that the spindle is all diffuse, all over the place. That the poles are, in one case, bigger, in another case, they're amplified. And if you look at the DNA, it's not aligning at all. And so you have these very ugly-looking mitotic figures. So the problem is that overexpression of PIM is correlated with this genomic instability, and it's been demonstrated to occur in a passage-dependent manner. So what do we mean by passage? Down here we're looking at passages. So you grow cells in a petri dish. The cells grow until they're confluent, or until the cell is completely filled with cells. So you take the cells, you split them, and put them into two new petri dishes. That's called a passage. So in this case, the cells were passed 60 times. And in this axis, we're going to look at the percentage of cells that have extra chromosomes. So if we look at normal cells, we don't do anything to them, we just passage them along, we see that there's no increase in the number of cells that have extra chromosomes. if we take the inactive, the kinase dead PIM, insert it into the cells, and pass them 60 times, again, we see almost no increase in the percentage of cells with extra chromosomes. However, if we put an active PIM into those cells and passage them, what we see is that by 60 passages, we have almost 90% of the cells have extra chromosomes. So the probability of coming down with cancer, if that was going on in your body, is pretty good. So how might PIM be involved in the buildup of this genomic instability or this aneuploidy? By allowing cells to survive when they have abnormal chromosome numbers that should trigger cell death, something we call programmed cell death or apoptosis. So what is apoptosis? Why is it good? Well, let's say we take a cell here, and it's either got too many chromosomes or it's got severe damage to its DNA. And that triggers the apoptotic pathway. So the cell starts to disintegrate from the inside. And it starts breaking up into small pieces. And those small pieces are phagocytized by the phagocytic cells, which are the cleanup crew in your body. And it doesn't affect anything else. The only thing that happened is that one cell died. Now, we have another kind of cell death called, and this is gentle. This is a very gentle way to knock off cells. Necrosis is another form of cell death. And in this case, it's a catastrophic cell disruption event. And the cell essentially blows apart, releasing all the internal contents into the surrounding tumor, and that results in inflammation. And this is an inflammatory response and can cause damage and can cause real problems. So on the next slide is a very good example of how PIM promotes cell survival by blocking apoptosis. Now, in the last 8 years, we've all, colleagues of mine and in our lab as well, have determined several pathways how PIM can inhibit apoptosis. And my last graduate student actually discovered this pathway. So when there's DNA damage, one of the things that happens is this kinase called ASK1 is activated. And when it's activated, that means it can now phosphorylate other proteins, and it will. And in fact, the dotted lines means there's a whole cascade of events that occurs. And it ends up activating the proteins that are involved in apoptosis. And apoptosis, of course, involves cell death. So if you have severe DNA damage, mutations, too many chromosomes, you really want this pathway to be activated because you want to get rid of that cell. However, what she found was when PIM is overexpressed, it can phosphorylate ASK1, and that actually inactivates it, even if you have mutations or too many chromosomes. It completely knocks out that cascade, which means you don't get apoptosis or programmed cell death. And as a result, that cell survives and now can carry on those mutations for generations forward. And it probably accumulate more mutations. So next we have an example of how PIM probably interferes with repairing DNA damage. This is a simple diagram of the cell cycle. The M down here stands for mitosis, and this is where our cells actually undergo cell division. These daughter cells then will enter the next phase, which is G1, and this is a stage where they prepare to synthesize DNA, which occurs in the S phase. In the G phase, they now prepare to go through mitosis. Now, if there is severe damage or just a little bit of damage that can be repaired, another protein called P21 is activated. And P21's job is to stop the cell cycle at this point and at this point. And so the idea is, in case there was any DNA damage here, you want to get it repaired before you duplicate those chromosomes by synthesizing DNA. If, in fact, that DNA damage occurred while you were synthesizing your DNA, you want to stop it here so that everything is repaired before you go through mitosis and spread that damage or those mutations to the two daughter cells. So the cell cycle inhibitor P21 looks like this. Actually, it doesn't, but this is a good representative. And it binds to another protein called pCNA, and it causes cell cycle arrest at the G1 or G2 checkpoints for repair. So here we see when P21 is binding pCNA, and it happens to be at this end of the molecule. And pCNA, by the way, stands for proliferating cell nuclear antigen, and it functions to allow efficient DNA replication. So if it's bound to P21, you don't get efficient replication, you get inhibition of the cell cycle, which you want. Now, it turns out, at this same end where pCNA binds to this protein, there's a consensus sequence that PIM likes to phosphorylate. And PIM will phosphorylate that. And when it does, what happens is it causes the dissociation of pCNA away from the P21, freeing it up, and that results in DNA replication to continue, and it reduces the efficiency of DNA repair. So to summarize, what makes a good target for PIM makes a good target for chemotherapy? Number one, the kinase is constitutively active. Number two, inhibiting PIM1 activity in normal tissue produces no detrimental effects, and we saw that from the knockout mice. 3, PIM continues to both cell survival and cell proliferation in cancer cells. And 4, inhibiting PIM in cancer cells leads to apoptotic death. And I haven't showed you that data yet, and I will in just a moment. And 5, PIM interferes with the DNA repair of DNA mutations, which I just showed you with the P21 story. Okay, 5 years ago, 2005, almost to the week, 4 different groups came out with the crystal structure of PIM. And this is because at that time already information was accumulating that said PIM may be very important to cancer production, and it may be the kind of a target we want. It won't cause, if you knock it out, it won't cause catastrophic problems to any other tissue. So, and the reason why they did this is because they wanted to know what small molecules, what kind of a molecule would fit into the active site of PIM or into the ATP site of PIM. So it turns out that since this time, a lot of progress has been made. There are 2 companies, Supergen Incorporated, that has identified a compound that has an effective IC50, which means kills half of the cells that are treated at a very low concentration, 50 nanomolar. And in fact, Valiant Pharmaceuticals also has a compound like that. Now, these are not the only 2 companies that are interested in kinases. Kinases are a big deal this day. It's a hot ticket in cancer research. And these are all the companies. You might even recognize some. There is, let's see, DuPont, and Pharmacia, and Pfizer, and Novartis, and Merck. And there are many more. I just didn't put them down there for you. So they all know, actually, that there's money in this. If they can find something that can actually stop and kill tumor cells without doing a lot of damage, they're really in the money. So this is the experiment I said I was going to show you and didn't have a chance to show you. This is looking at Supergen's SGI 1776 compound on leukemia cells, and these were taken from patients with chronic lymphocytic leukemia, and this was done in vitro. What they did was, this is a 24-hour experiment. They took the leukemia cells, put them in culture without anything or with the drug. 24 hours, they have almost 95% of the cells being killed off. So what happens if you put normal cells treated with the drug? And here are normal lymphocytes from normal individuals, and here's no drug, and here is drug. You notice that there is no real killing of the normal cells. So this says this is really great. It doesn't hurt normal cells, but it does kill cancer cells. And finally, the exciting news. There is a phase 1 clinical trial underway, and it was started in April of last year, and it will be completed in July 2010. And And this clinical trial is to determine the maximum tolerated dose of Supergen's SGI 1776. Well, I have to tell you, they've hardly published any of this data. So, and this is supported by NIH. So what this says is they took all their data, they've done probably animal studies, preclinical animal studies, they've done in vitro studies, and they have really good data, and they went, and this is now being underway. So in a phase 1 clinical trial, you recruit patients that have no other option, and what you're looking for is to see how much of whatever it is you're going to use you can get away with without doing damage. And the patients that were recruited to this particular clinical trial were those with hormone and Taxol refractory prostate cancer. In other words, nothing is working for them, so this is their last chance. Maybe with a little bit of luck, this will help them. And the other group of patients that were recruited to this clinical trial are patients with relapsed or refractory non-Hodgkin's leukemia. Again, they've gone the whole gamut. They've tried everything and nothing works, and so this is, again, their last hope. If this clinical trial is successful, they will go on to phase 2 clinical trial, which will actually test patients that may have more of a chance than these patients. So, in conclusion, PIM appears to be an attractive target for some cancers because inactivating PIM in normal cells does no harm, and that's a big biggie. 2, eliminating PIM1 activity from cancer cells is lethal. It kills them. But cancer cells require an extra help to survive and proliferate. And because several very specific and potent drugs have evolved that inhibit PIM activity. And so I should just mention about that SGI 1776 drug. It does not affect any other kinases. That's the beautiful thing about it. And also, PIM appears to be an attractive target for diagnosis and also prognosis for some cancers because the levels of PIM correlate with the stage of malignancy. So, not any one person could ever do all this stuff. I just gave you a condensation of 20 PhD theses. So, it does take people working together to do remarkable things. So I have acknowledgements. These are people that have actually contributed or worked with me on PIM here from WSU, and I should mention that Zeping Wang probably has done more than almost anyone because he's had his hand in almost all the experiments. Nationally, all my colleagues, and then it turns out I've even worked with more people internationally. And then I have to acknowledge my graduate students and postdocs all have contributed to the PIM story. And last but not least, my husband. And I have to thank him because he's the one that actually was the first one to clone the entire PIM gene. Anton Burns really only cloned the cDNA. And PIM not PIM. Ray cloned the entire PIM gene, including the regulatory regions. And so that's what I use in my research. Now, you know you cannot go home at night and not talk about science occasionally, right? And so we talked about PIM, and he gave me suggestions, and we talked about what good things to do. So I really owe him a lot because he did help me very much. And then I have to say that the mouse got the cheese. So thank you.
I'll take a couple questions.
Oh, sure.
If we have time for a couple of burning questions, if someone would like to ask something. There's one right back.
Oh, yeah, okay.
Yes, I was thinking, the PIM as a marker for certain kinds of cancers, especially prostate cancer, kind of suggests that perhaps it's upregulated in a targeted fashion.
Well,
I guess the question would be, why would PIM1 genes be the target of
okay.
regulation?
So that was the other 5 theses I didn't talk about. So it turns out cytokines induce it. Hormones induce it. And so one of the biggies that induces is an IL-6. And IL-6 is a pro-inflammatory cytokine, and that elevates it. And it turns out, if you look at the literature, that individuals as they age, and that goes for all of us, actually we start having higher and higher levels of IL-6. And so there's a connection. That signals the JAK-STAT pathway, and the STAT transcription factors upregulate PIM by gangbusters, as does NF-kappa B and some others.
Someone else, right here.
Yeah.
Nancy, I appreciate diagnostics, as you know. And since PIM is basically ubiquitous, how will you detect it? How can it be a diagnostic? Is it serum-based? Is it tissue-based? And what kind of patient do you go after to detect it?
Okay, so the question is, how can you use PIM because it's ubiquitous? And where would you detect it, in the tissues or elsewhere, right? Well, it turns out that other things I didn't tell you about because of time, PIM has a very short half-life. So in normal tissue, you really don't see it. Where you see it is when cells are activated to divide, it's there for a short period of time during the cell cycle. So you would not see it normally outside of cells. I mean, the half-life is like C-Nick. It's like 15, 20 minutes. But under certain conditions, it will be stabilized. And so in cancers, that's what happens, is that the message and the protein itself are both stabilized. So the half-life is much longer, up to an hour and a half. So you would have to look at it with antibody in tissue sections.
Okay, I want to thank everyone for attending the lecture today, and I want to especially thank the committee that was the selection committee for the Distinguished Faculty Address, chaired by Ken Cassavant, and Alex Tan, also a member. And there will be a reception immediately following this across the hall in the dining room. And let's thank Nancy again for an outstanding lecture.
All right.